From Microbe TV, this is TWiEVO, This Week in Evolution, episode 22, recorded on August 30th, 2017. This episode is brought to you by Blue Apron. Blue Apron is the #1 fresh ingredient and recipe delivery service in the country. See what's on the menu this week and get 3 meals free with your first purchase with free shipping by going to blueapron.com/twee. Hi, everybody. I'm Vincent Racaniello, and you're listening to the podcast on the biology behind what makes us tick. Joining me today from Salt Lake City, Nels Elde.
Hey, Vincent. Great to be with you again from Elde Lab Studios here in the foothills of the Wasatch Mountains.
I'm not in any foothills here. I'm just on an island on the East Coast.
Yeah, but you got a nice view across the Hudson River.
Got a great view. I'm not complaining. It's 26 Celsius here and mostly sunny. And, the humidity's gone, which makes me sad, but that's the way the seasons go. How about you? You have nice weather there?
It's warm. Summer is holding on.
Good.
Holding on strong. So let me see if I can dial it up. We are a little bit north of 90 degrees Fahrenheit at the moment.
Wow. Maybe I should come out there.
Yeah, and no humidity as well. It's pretty good.
I happen to like humidity. I don't know why. I just love walking out in the morning, getting that blast of moisture.
I hear you.
Must be my Mediterranean genes.
There you go.
We have a guest today, don't we, Nels?
We do, a great one.
Now, this is someone you know, I understand. She is in the Department of Genome Sciences at University of Washington School of Medicine, which is even farther west than you are, Nels.
That's right.
Maitreya Dunham, welcome to TWiEVO.
Hi there. Thanks for having me on.
My pleasure. So you guys know each other, right?
Yeah,
we know each other from when Nels was in Harmeet's lab.
Did you overlap?
Well, so Maitreya, when did you start? You started your lab before I got there. I
started at UW in 2008, long time ago now.
So give us a little bit of your history. Where are you from originally?
So I grew up in rural Tennessee and then have been living kind of the migrant scientist lifestyle since then, bouncing from coast to coast a few times. I did a biology degree at MIT and then did my PhD with David Botstein at Stanford in genetics, and then became one of these independent fellow gigs at Princeton, which was a great kind of way to get started with my lab, and then moved to UW for my professor gig.
So Maitreya, remind me a little bit about the sort of super postdoc that you did at Princeton at the Lewis Siegler. So you were running your own group in essence, right?
Yeah, it's really a great deal. So it was right out of grad school, and I was given resources to start my own small lab that was mainly run on undergrad power, along with some really fantastic technicians and some visiting professors who came through. And it was just a brand new program at that time. They had just started it, and me and Will Rue, who's now at Toronto, were the first 2 fellows. So it was really fun to be able to kind of choose our own adventure for that. And the one unique thing about the Lewis Siegler program is that there's a lot of involvement in teaching. So we also got to help develop the integrated science curriculum that Princeton was in the process of developing at that time.
Oh, so I get it now. You went to UW 2008, you said, right?
Right.
And Nels was there as a postdoc with Harmeet, right?
Right.
Correct.
So that's how you know each other. Got it.
Yep.
Okay. Very good. Well, you have zero accent. Is that because you've been bouncing around a lot, or do people from Tennessee don't have much of an accent to begin with?
Oh, my parents grew up in Michigan, so
I see.
I think that might. And we lived out in the woods. Maybe I just never picked it up from.
Did you go to high school in Michigan?
No, I went to high school in Tennessee. I've been in
Tennessee
since I was like 6 months old. So
it's quite. You have a Michigan accent. I don't know, but certainly not a Tennessee accent. Well,
nope.
That's good. I don't know. It doesn't really matter, but it's just curious.
I get it a little bit when I go home and visit. You kind of pick it up.
Yeah, that makes a lot of sense. Yeah, exactly. Well, we have you here today because we wanted to talk about this paper that we recently published, which Nels picked up. So this was published in June of this year, and it's in the journal Genetics, and the name is Experimental Evolution Reveals Favored Adaptive Routes to Cell Aggregation in Yeast. And the authors are Elise Hope, Clara Amorosi, Aaron Miller, Colina Dang, Katie Smukowski-Heil, and Maitreya Dunham. Did I massacre any of those names, Maitreya?
No, you got it.
Well, who are the people who's the first author and so forth? What's their roles in your lab?
Oh, yeah. So this was really a great joint effort by a lot of different people in my lab that really came together nicely. So the project was started by Aaron Miller, who was a graduate student in my lab, one of my very first graduate students, actually, and is now at Zymergen. And he had done a large series of evolution experiments. And as a side effect of that, we came up with these aggregating mutants, and they'd always kind of been in the back of the freezer, and Elise adopted them, and we ended up involving an undergrad, Colina, and my teaching at Cold Spring Harbor's yeast genetics and genomics course. So we sequenced several of the strains every summer for the last few summers at the course. And Katie was actually a TA 1 year at the course with me when we figured out one of the big interesting mutations that we focused a lot of the paper on. So it's kind of come together from multiple people contributing to it over the years.
And maybe, Maitreya, tell us a little bit about that yeast course that you've been involved with. Didn't you just finish that up for this year?
Yeah, that's right. We just finished. This is the 6th year that I've taught it. And it's really like a yeast boot camp, pretty much. Cold Spring Harbor has this great history of courses going back to, the phage course, right? So it's 3 weeks. Super intense. We do 11 different labs, and students come in from all sorts of different backgrounds and want to learn yeast. And so we teach them all the basics, and it's kind of our aesthetic is to kind of ground it in the classics, but do things in a modern way. So we do whole genome sequencing. This year, actually, I developed a new teaching lab on deep mutational scanning, which is a way of doing saturation mutagenesis and quantitatively measuring the effects of lots of mutations at once, which mostly worked. Most made it. It didn't quite go completely as planned, but nothing quite ever does. But it's a lot of fun. It's a really great opportunity to kind of inculcate the next generation with yeast, and also to do some science. So every year I try to bring some real experiments so that things aren't total cookbook and where the results. And I think it makes it more exciting for me, certainly, and gives the students a sense of discovery and engagement.
Yeah, sounds great. Vincent, what do you think? Are you going to sign up next year?
I don't know. I'm too old.
We have professors take it every year. It's really fun, actually, including a lot of professors who want to pick up yeast for teaching. So we've had several people take it over the last few years who are introducing yeast into undergraduate labs or things like that, in addition to people using it for research purposes.
You may not know this, Nels, but many years ago, we worked on yeast for a bit. In fact, we used to have some yeast people here, and Maitreya, you might know some of them. We used to have Aaron Mitchell.
Oh, yeah.
We used to have David Shore. Lorraine Symington is still here. Rod Rothstein is still here. But David Shore and Aaron Mitchell have left.
Right.
Oh, also Marion Carlson is here, you may know.
Oh, of course, yeah.
Didn't she work with Botstein?
She did, yes.
That's her. Did you guys overlap?
Academic sisters.
Okay, there you go. So she's still here, although she doesn't have a lab anymore.
Right.
But she's still around. But we worked on yeast to try and... So we work on viruses that have internal ribosome entry sites for translation, and we had stuck those into yeast to try and identify, genes that are needed for the activity of those sites.
Right,
so
right.
We did that, and I still have a freezer. We bought this mutant collection of like 4,000 strains, I think, and we went through each of them, put the plasmid in each one of them. And these are the old days before you could do any kind of big data collection. Right? Do it differently now, but we still have these strains. I don't know what to do with them because we don't do it anymore. But there's so many courses I'd love to take at Cold Spring Harbor. They're so cool. right?
Yeah, we'll have to put a link to the yeast course
for sure
for the
that'd
episode
be great.
notes here. Yeah. So in Maitreya, isn't there also like a famous yeast plate race that happens? How'd that unfold this year?
Oh, sadly, we did not win this year. We lost to Gene Expression, who are our kind of constant competitors in this. But the plate race is we save our plates the entire summer in the yeast course. So this is hundreds, maybe thousands of plates by the end of the summer. We make them do a lot of Tetrad analysis, so we go through lots of plates. And the race is a relay race where you have to carry a stack of 40 plates and run a circuit that's right in front of the Delbrook building. And Cold Spring Harbor shuts down the road. All these people come. They have an official photographer. It's crazy. And this year we had a tragic drop where if you drop your plates on your circuit, you have to run back to the start and get a new stack and then run your lap and then hand off your stack to the next runner. So
I've seen those pictures.
Oh yeah, they're hilarious. Really this year, actually, Cold Spring Harbor made a promotional video about it. So maybe I'll dig that up and find out where they put it on their website.
I always wondered what that was. I passed it in the hall and I said, what are those people doing? Now I know.
Oh yeah.
That's pretty cool.
Looks like fun.
No, the course is a little bit like science summer camp. So at the time, it's extremely exciting and you get really into it. And then afterwards, you're like, that was kind of hilariously silly, but it's great fun.
No,
Sounds perfect.
It's a lot of fun out there doing all sorts of things for sure. And there are other great courses there as well, of course, that have some of them gone on for many, many years. So great tradition out there, right?
Oh, yeah.
I wanted to ask you, we're going to ask you some questions that may seem like we should know the answers, but we have lots of listeners that aren't scientists actually, which is great.
Oh, that's great.
So first, maybe you could just talk about experimental evolution, what that means and what kind of things you can do.
Oh, yeah. So one of the tools that we use a lot in my lab is trying to watch evolution as it happens. So what you can do is you can take, a single cell of yeast and you can start a culture with it and keep that culture growing for months and months of time, hundreds of generations of yeast culture time, and allow the cells to have mutations and compete with one another. And the main selection pressure that we use in the lab is typically nutrient limitation. So we'll take their favorite media and we'll reduce the concentration of one of the nutrients until the yeast are essentially competing over who can best assimilate that nutrient. And it sets up a really clean selection pressure, which is a lot different from trying to understand kind of historical evolution, right? You don't really know if you go out into the wild and isolate a yeast cell, you don't really know what it's experienced or what its ancestors have experienced. So it can be difficult to link genotype to phenotype to figure out what mutations in its genome actually matter for adapting to whatever conditions it's encountered. So experimental evolution allows you to really simplify that process by defining the evolution conditions, you can also save like every single day's culture so that you have a complete historical record in your freezer, which allows you to kind of go back in time. The other important thing is it lets you repeat the experiment many times. And that was one thing that was really beneficial for this paper that we're talking about today, is that we were able to actually compare how different strains evolved by doing dozens of evolution experiments with the two different genotypes and getting high enough numbers that we can actually compare them with some statistical accuracy. So it allows you to do experiments on evolution in a way that is difficult to do in kind of more natural settings. Now, the downsides, of course, are that it is so simplified, right? It is really, really overly simplified. And in fact, the method that we use is a continuous culture gadget where we're constantly flowing media in and constantly flowing media out so that the cells don't even see any fluctuations in conditions as much as possible. And even that simplified environment, we're always constantly surprised at the complicated things that the yeast will do, even in that simplified scenario. So eventually we'll try to get more complicated, but I don't think we've yet kind of maxed out on the types of questions that we can ask using this really simple system.
Yeah, and just maybe to back up for a second. So one of the reasons I'm really excited that Maitreya is with us today is that on TWiEVO so far, we've really focused a lot of attention on kind of comparative phylogenetic approaches, or retrospective evolution. And what a great example of prospective evolution or experimental evolution, as Maitreya is saying, seeing these events in real time. And for me, what's really fun is that, I think it's easy for us to think about this in things like viruses and microbial pathogens. But the fact that even the eukaryotes, in this case yeast, you can see evolution happening on a time scale fast enough that you can actually do experiments like this.
This is the sort of thing that Lenski does as well, right? With his bacteria, growing them for decades, I think, in his case, right?
With E. coli.
Oh, yeah, no. Yep. The Lenskiites, the type of experiments we do in yeast are a blink of the eye to the time scales that they've done. So there are people from his group who have gone on to do similar long-term experiments with yeast. So Cliff Seal, for example, has done several thousand generations of passaging for yeast in the kind of same back dilute serial dilution regime as what they use. Let's
talk a little bit about the machine, if you will.
Oh yeah.
This, which is a Chemostat, which you mentioned briefly, where you keep the yeast in and you flow media through. You actually have a picture of it, I think, on your website. It's just to remind you, there are lots of white tubes. There are 1, 2, 3, 4, 5 shelves, and some different colors, which I guess are the caps. Can you explain that to us?
Yeah, sure. So this was really key to the type of work that we do now, because when I first got started in this area, we were using these elaborate glass-blown, really beautiful pieces of equipment, but which were really difficult to do lots and lots of. First of all, they took up a lot of space, and you were kind of at the mercy of scientific glass blowers to make them for you. And so when I started my lab here, we decided we needed to miniaturize it. And so what the photo is on my website is our what we call them the mini stats. And it's essentially the same plumbing. All you need is a media inflow and a media outflow and a way of mixing and aerating the cells. It's literally made out of test tubes and needles and pump tubing and aquarium pumps. The fanciest piece of equipment in it is we did buy a nice peristaltic pump to allow us to control the media flow pretty accurately. But you can run a whole setup of 32 of them at a time and build them yourself in your lab for on the order of $10,000, which is, as these things go, not so bad. I've been really surprised. If you are really excited about them, you can watch our 20-minute instructional video that we put together on how to build them. And I've heard from dozens of people around the world who have built them. We occasionally host people in our lab for what we fondly call Chemostat camp, where you can come and learn how to build them. But between the 20-minute instructional video and we have a pretty detailed set of user instructions on my website as well, people are able to build them themselves without much help from us. So it's been really exciting to see that it kind of fills a niche in the community and to see how many people are using them for different things. And it's really allowed us to really up the number of evolution experiments we can do and get those replicates that really help for trying to understand things.
Wow, it's really cool. How many mini stats would you estimate that you have up and running in your lab?
Oh, let's see. The lab record is 128 at once.
So let me understand. It's basically the yeast are in a tube and media is being pumped in and out. What's the flow rate roughly?
So the flow rate is about a generation time takes about 5 hours. So it's slower than yeast would normally grow in kind of a flask, given maximal conditions. But by controlling that growth rate, we can kind of allow them to have very steady conditions.
And the media is coming in very slowly?
Right. It's drip by drip.
Now, these experiments go on for some time, but don't the yeast eventually overgrow the culture and die? And what happens with all that?
Well, that's the great thing about the constant flow and the constant overflow, is that every 5 hours, you're getting a media replacement. So you're essentially diluting out half of the culture and keeping half of the culture all the time. So you always have new cells growing and populating. And if you aren't a growing cell, then you're quickly going to be outcompeted by your neighbors.
Oh, so the dead ones get pulled out with the medium then, is that it?
Right. There's this constant inexorable dilution.
Cool. That's pretty neat. I think Lenski just grows his in flasks, right? So
that's right.
It's harder. You have to spin them out or whatever he does. I don't know.
Oh, I mean, it's actually pretty easy. I think the flasks are nice because it is very straightforward. You just pipette some of the culture into the next day's flask. The thing that I think is mainly different is that if you're in a flask, then you're experiencing different nutrient conditions throughout the day. In the morning, things are good, right? You've got all this nutrient to eat, and then you grow as fast as you can to eat all that nutrient. And then you run out maybe in mid-afternoon, and you have to spend the rest of the day in stationary phase or whatever, waiting for that next day's back dilute to happen. So you can get specializers. You can get things that are fast off the marks in the morning, things with higher maximal growth rates, things that divide a few extra times even when they're low on nutrients. Which I think is interesting, and really, obviously, they've made great work with that system. But in the Chemostat, you don't have any variations like that. Because you are constantly diluting in and overflowing out, the culture reaches a steady state very quickly and then stays there for months. So you can completely control that selection pressure.
I guess you could use this for mammalian cells as well, right?
Oh, sure. So one of the devices that we used to use before we built the little mini guys is a fermenter that is sold for use with mammalian cultures. Everything goes more slowly. And of course, yeast are pretty good at outcompeting any stray contaminants that might creep in, whereas mammalian cells are a lot harder to keep happy. So I think there's definitely some promise there, but no one has really made it work in a really great way. There are people doing like more kind of batch transfer, sort of just, serially plating. And of course, people have done it accidentally, like what the heck HeLa cells are now versus when they were isolated is kind of fascinating.
Hey, Nels, what do you think of putting mammalian cells in and then throwing in some viruses?
Well, yeah, I like it. And
long term.
Correct. I have to confess that I'm super jealous both of the yeast setups, the chemostats, even the flasks, because as in my lab, we do some experimental evolution with vaccinia virus in particular.
Right.
And to do that, we need to grow dishes of mammalian cells, infect them, and then the viruses run their way through there. And then you have to pull them off, harvest the viruses tighter and kind of do it. And so the amount of labor that you can bypass with these single cells organisms that are independent is not trivial at all. But yeah, maybe taking a little bit of a hint from some of these great advances, we could establish something a little more automated in the future.
I think you need to sign up for a Chemostat minicamp.
Yeah, boot camp, absolutely.
Boot camp and adapt it to mammalian cells, because that would be cool to look at long-term virus and cell evolution in this kind of culture, right?
Totally agree. Yep.
Yeah, and there are people doing that in bacteria, where you can do bacterial and phage coevolution in chemostat conditions. So it's a little tricky to get the conditions just so that you have a stable setup, but there are definitely models out there for that.
So Maitreya, set up for us the question you were trying to address using these chemostats.
Okay, so I just made a whole big deal about how the selection pressure is something that we can completely control, and this whole paper is about how that's not entirely true. So one of the side effects of growing in this continuous culture device is that, you get what you select for, right? If we're selecting for genotypes that have longer that last longer in the culture versus all their neighbors, then one way of doing that is to, get better at using the limiting nutrient. But another way of doing it is to stick to the walls or to make a big clump and sink to the bottom away from where our overflow port is.
Yeah, of course.
They're clever, right?
Absolutely. Things you would not think of, right? Yeah.
Right. But it's actually been kind of a bane of chemostats' existence since they were developed back in like the 40s and 50s. Everyone noticed immediately that they would accidentally select for clumps and biofilm formation. And it's been one of the main limitations for chemostats for a long time, because, you can't do these multi-thousand generation experiments like you can in flasks, because you nearly always eventually select for these biofilm and clump traits. And yeast are particularly good at doing that, it turns out. If you go out into the wild and isolate yeast, you'll find that they don't grow necessarily like unique single cells that float around planktonically separate from one another. They, in fact, will grow as clumps. They'll grow as communities. Their colonies, for example, often will have beautiful structure to them as opposed to the kind of, flat featureless colonies that you're used to seeing with lab yeast. And it turns out that the lab yeast that we normally use actually has a mutation in it, which makes it not do this clumping trick that its wild friends would normally do. And while that's good for microbiology, it might not be so good for the yeast. So the yeast and the chemostat have kind of rediscovered their ancestral selves in some ways.
So this study sounds like it was a case of taking lemons and making lemonade, that you got these sort of nuisance features or phenotypes, but you were able to kind of turn the tables and actually learn some interesting biology.
Well, we also were finally tired of getting it over and over again. We were like, can we stop them from doing this? Can we stop evolution? Right? Yeah. So that was part of the incentive, was to see if we could use evolution to stop evolution. Could we, by understanding what mutations they get to develop this trait, figure out a new strain background that we could engineer to be more resilient to evolving this trait, that where we could evolve it for longer without it actually developing this trait.
So maybe let's talk about these traits in a little more detail. So you've kind of mentioned aggregation or biofilm formation. There's also this idea of flocculation. Can you just maybe define some of these terms?
So flocculation is where two yeast cells can stick together via nanoproteins that are on their cell walls. So it's kind of like cell Velcro. And it's those cells can it's actually reversible. If you chelate away ions, you can make them disperse, and then they'll come back together again if you rinse them again. The other way that cells can aggregate is by a mother-daughter separation defect. So this is budding yeast, right? So mother cell will make a bud, and there are particular mutations that you can get that make that bud not separate properly. So it doesn't undergo cytokinesis properly and will stay stuck to the mother. And you can make these kind of chains. Other labs that work on this have called them snowflake yeast, and have been studying that trait as a type of multicellularity, perhaps. So we got both of those mechanisms when we looked at who had evolved in our chemostats, but largely we got flocculation.
Go ahead.
It sounds like you had something in mind, so go ahead.
Oh, so I was just curious a little bit about how you made this selection. So as I started to read the paper, I thought, and as you've actually, in some of our conversations before, you mentioned that the yeast will start growing up the tubes onto the walls, and so I was sort of assuming that you were just scraping the yeast off the wall, but it sounds like that's not the case. That you were doing a different aggregation assay as these things are collecting and not overflowing as they usually would if they were dispersed. Is that right?
So we did two things. One was that whenever we would decide the experiment's over, we would check the tube and see if there were visible clumps in it. And if there were visible clumps, then we kind of fish them out with a pipette and freeze those separately from the rest of the population. We would also try to scrape off whatever was on the wall. There's this phenotype, we call it the bathtub ring, where you get this like aggregation of yeast all around the top of the tube. And one thing that they're very clever about is once they kind of develop that ability to stick to a surface, then some of them will eventually colonize the input port that delivers the media. So we're bubbling lots of air through. So I'm imagining there's like aerosolized yeast kind of in the headspace of the chemostat. And so eventually somebody lodges up in the media feed, and then they're a real winner, right? They get the media before even it gets delivered to the rest of the tube. Wow. So that's something we constantly are on the lookout for. We have to swap out the needles and all this stuff. It's a huge hassle.
Mm-hmm.
So what is the starting point for this experiment? What's the strain, and what are the different growth conditions that you ran?
Yeah, so the experiment started out as just we'd like to understand what mutations allow yeast to adapt to different nutrient limitations. So we have 3 that we use for a lot of the experiments in the lab. They're sulfate limitation, glucose limitation, and phosphate limitation. So Aaron and a team of undergrads evolved 96 different cultures that were under these conditions. And we found, to our dismay, that about a third of them developed some sort of clumping phenotype or wall growth phenotype by the about 300 generations. And those strains end up being kind of annoying to do crosses with, or to even to isolate clones. You know, you're used to being able to streak for single colonies and know that that colony derived from a single cell. But if the cells are all clumped together, they violate all these rules, right? So Kalina ended up having to go in with a micromanipulator and pull off a single cell from each of the clumps in order to clone purify all these strains that we eventually sequenced.
Now, if you did this without any nutrient limitation, would you not get any kind of clumping or biofilm formation?
Well, the selection pressure for this is simple residence time in the chemostat, right? So I think any sort of a flow device, you would select for things that can stay in by physical means as opposed to by kind of competing over resources.
And is there a timing difference if you compare, full media versus nutrient limitation? Does the clumping happen faster without nutrients compared to the complete medium?
So we have never tested that, and that's because in the chemostat, you pretty much are forced into a nutrient limitation.
Okay.
Whatever runs out first becomes your limitation, so you can either control that or not. So so instead, what we did is use different nutrient limitations, and it is definitely the case that in some conditions we saw more and faster clumping than in other conditions. So glucose limitation, for example, seemed to elicit more frequent and earlier clumping than our other nutrient limitations. And that's actually something that is an unanswered question that we have not figured out why that is. Whether there's something about the clumps that are kind of co-adapting to the nutrient limitation, or whether it could be that it's a fitness distribution issue where in sulfate limitation, for example, we know that there's this really amazing gene amplification that has a very high fitness consequence. And glucose limitation just doesn't have such a mutation. And so it could be that more kind of compressed range of beneficial mutations kind of lets the clumping mutations be more of a contender versus the other limitation. We haven't actually figured that out yet. It's still an interesting question.
Yeah. So, and before we get into some of the results from all of your whole genome sequencing, so a lot is actually already known, isn't it, about the genetics of cell aggregation or clumping in yeast? So there are these flow genes. Maybe help us with a little background on that.
Yeah, sure. So, even though so we started out with this lab strain that is usually not used for studying cell aggregation because it, like I mentioned, that has a mutation already in it that prevents this behavior. It's a point mutation in the main transcriptional regulator of this whole pathway that controls all these flocculation related genes. So, like you say, they're all called flow genes, which led to the article got posted on the Saccharomyces genome database, and they made all these flow jokes about it was kind of hilarious. But those are some of the genes that had already been known, and there have been a bunch of different labs that have been studying this pathway in various ways. Jerry Fink's lab in particular is one that we've followed his work very closely, but there's a lot of different labs that have worked on this. Kevin Verstreppen is another one who's worked on some of these genes and their evolutionary consequences. But there's also a lot that's unknown because some of it seems to be strain background dependent, and a lot of it is studied in only a limited range of strain backgrounds. So we thought that maybe there were still genes left to be studied that hadn't been necessarily found yet by the screens that had been done already. But there's been some nice quantitative trait analysis work, some nice genetic screens, lots of different approaches. So we definitely had a basis on which to start, which was great.
Now, one of the things you mentioned very early on, you say you got a third, by 300 generations, about a third had gained a visible aggregation phenotype, which was not present in a strain that has a nonsense mutation in flow 8.
That's right.
So what does that mean?
So that's that ancestral strain. So it starts out with that nonsense mutation, and that prevents it from doing the flocculation. So the starting strain is not able to make clumps, but the evolved strain, which only has a handful of mutations in it relative to the ancestor strain, is now able to make clumps, sometimes very robustly. So that's what we set out to do, is to figure out what are the mutations that let you start from a non-clumper and end up as a clumper.
Okay. And at this point, we don't know if those are simply changes in flow 8 or something else, right?
Right.
Because you can't tell just by looking at the aggregation phenotype, right?
No. So that's what we were curious about, is how many ways are there to arrive at this phenotype?
Okay.
Yeah. And so then if you look at figure 1 of the paper, you have a pretty stark and clear visual demonstration of the difference between the ancestor, just these nice photographs of the test tubes, and then all of these, was it 23 or so evolved clones that you ended up streaking out? And you can really see that these things are collecting at the bottom of the tube relative to the dispersed ancestor. And so remind me, Maitreya, was there, we mentioned this difference between mother and daughter sort of separation versus the clumping, the Velcro sort of style connections. And what was the distribution among those 23 clones of those various types of phenotypes?
We only got a couple that were mother-daughter separation, and the majority of them were flocculation.
So this flow 8 is is it a nonsense mutation?
It is, yeah.
And what is it?
So that's
is that
one interesting thing. We never saw a reversion of the flow 8 mutation.
It's amazing.
Isn't that cool? And we never found an informational suppressor of the flow 8 mutation either.
I'm amazed. See, if I had known that and I wanted to do this experiment, I would say no, because it's just going to be reversion of the flow 8. And I wouldn't do it because of that. So it goes to show you, don't worry about it.
Yeah.
Well, it's actually still an open question to us. So we tried fixing the flow 8 mutation just because. So the experiment that had been done in the Fink lab was to put the wild type flow 8 allele on a plasmid and put it into that yeast strain and show that you could recover flocculation. So we thought reversion would also be an issue.
Yeah.
But when we changed that exact codon on the chromosome, we actually didn't see a full recovery of flocculation. And this is not in the paper. This is an experiment we've done lately, and that hasn't really been replicated. So we're not entirely sure that that's true or not yet. But it seems to imply that maybe the lab strain hasn't just lost the regulator flow 8, but it might have additional mutations in its background that limit its capability to develop this trait.
Is it possible that some of the aggregators you identified are fixing those mutations?
It's a possibility. So we think we understand most of them, but there are still a few that we aren't really sure the mechanism of action.
Okay. All right. So you had how many flocculating? 23 populations that are aggregating, right?
Right.
And
21 of those were flocculant.
Right. And so what was the next thing to do?
So we wanted to figure out what the causative mutations are. And this is one of those cases where we sequenced their genomes and then you start telling yourself stories, right? You're like, okay, well, there's all these flow genes, right? How hard can it be? We'll just sequence them all, figure out what the mutations are, and it'll be easy. And instead, what we ended up with was a list of mutations, many of which we could kind of come up with vague connections to flocculation, but it wasn't really a slam dunk in terms of, we didn't get that flow 8 reversion that would have been a smoking gun. So we turned to genetics to make sure that we understood causation. And this is a really nice approach. And this is also what I was teaching in the course. So the basic concept is that you use genetic linkage to zoom in on which one of the mutations is the right one. So you sequence a whole genome, you find out that it doesn't usually have just one mutation. It usually has maybe 4, let's say. Now, 3 of those, and also you don't know how many of those mutations do cause your trait. It could be that the trait is complex, right? And that multiple mutations combine to cause it. So the genetics also tells you how many mutations are you looking for. So all we did was we took each of the evolved strains and we back-crossed it to an ancestor of the opposite mating type. And then we sent them through meiosis and looked at how the trait segregated in the progeny.
So, and this is where you did some of this Tetrad analysis that you
That's right.
mentioned at the Cold Spring Harbor course. Yep.
Right.
It's good to have a lot of hands to do that, isn't it? To dissect out.
Yeah. So it was a lot of tetrad dissections for sure. And Kalina, my undergrad, was one of the she's a great dissector now. She did a lot of those in addition to other folks in the lab.
Let me ask you a bit about the whole gene. So you did whole genome sequencing, right?
That's right.
On each of these. So roughly how big is the cerevisiae genome?
Oh, yeah. It's only 12 megabases.
Okay. 12 megabases. So that's good. I have to point out that I've been on so many PhD student qualifying exams where you tell them, here, I want you to design an experiment to answer this question, but I don't want you to do any genome sequencing because that would be so easy, right? Now here, you are kind of contaminating them because that's what you did, although you had to do other things as well. I just think it's funny that we can do this and get an answer. Now, when you compared these 23 whole genome sequences, you might have already mentioned this, but I'll ask it again. How different was each genome, one from the other?
Yeah, so they only have a few hundred generations to accumulate differences, and so it's really only a handful of mutations.
Wow.
I forget what our maximum was, but usually we're sifting through something like 3 to 5 mutations.
Wow.
And among those mutations, so I think it's we commonly think that the most common mechanism is that you'll get a single point mutation, right? When you're replicating the DNA, that there'll be a mistake or something like that. But in fact, it's a collection of a lot of different mechanisms. So copy number variation, or you mentioned with that sulfate case where you can get aneuploidy or whole like chromosomal modifications or indels, those kind of things. Do you have a sense from this experiment or related ones, sort of what's the frequency of different flavors of mutations? Or is it all over the map, or does it depend on the selection?
Oh, I think that's one of the really interesting things about this project was we initially had only called SNPs. And at the course, it's also just easier to deal with SNPs because the data analysis pipeline is straightforward, whereas calling things like copy number variants and transposon insertions is just slightly more complicated. So we'd focused in on the SNPs initially, and several of the groups had no mutation. So they were like, well, I guess it doesn't have a mutation and sequencing doesn't work. Which was like, but you can see before your very eyes the segregation pattern, right? You can tell that the trait is segregating like a single mutation in the backcross. So there's a mutation there. So the other explanation is that sequencing just didn't find it, or that our data analysis pipeline didn't find it. And so it turned out that about half of our mutations were transposon insertions. And so we had to use, a little bit extra analysis to go looking for those.
Why wouldn't the sequencing pick that up?
Oh, no, the sequencing totally does pick that up. It's just that basic SNP calling pipelines don't pick up transposon insertions.
I see. Yep. Interesting.
Yeah, so this bugged me so much that some of the groups couldn't find their mutation that I ended up finding the first one literally just by looking at the sequencing coverage data for candidate genes.
Oh, wow.
So I was looking at flow 1 and noticed that. So what's nice is that what you do is you don't sequence the clones, you sequence pools of segregants, right? So you pool all of the segregants that clump and you pool all the segregants that don't clump. And then you should see perfect difference at the causative locus, but all the other loci that don't matter should be in both pools. So it really lets you tell apart who's causative versus who's just there for the ride or is, selected for by virtue of its role in nutrient limitation or something like that.
Neat.
And so I was able to find that transposon insertion only because there was a low coverage at the site that it went in. And then Katie, who's one of the other authors on the paper, she has been working on transposons as a separate project that she's still doing. And she realized that they were all multi-mapping reads, which indicates that there's a repeat element. So that's when we went and looked directly for transposon junctions and found them.
Cool.
Well, before we talk about that and the other finds, let me take a break here and tell listeners about the sponsor of this episode, Blue Apron. Blue Apron is the #1 fresh ingredient and recipe delivery service in the country. And what they're trying to do, their mission, if you can put it that way, is to make incredible home cooking accessible to everyone, and at the same time supporting a more sustainable food system, setting high standards for ingredients, and building a community of home chefs. If you don't like cooking, I personally don't mind cooking, but I don't like shopping. This will take care of it. You can learn how to cook, because what they do, for less than $10 a person per meal, they will deliver to you seasonal recipes along with everything you need, all the ingredients pre-portioned, so you can make delicious home-cooked meals in 40 minutes or less. You get this lovely recipe card that gives you step-by-step directions. Everything comes in exactly what you need, so there's no waste. You don't overeat. And if you follow this recipe, you will learn how to cook. And of course, you can keep the recipe card and then replicate it as many times as you want, because Blue Apron doesn't repeat their recipes every year. And you can customize your recipes every week. There's no obligation how much you have to buy. If you like meat or don't like meat, you can tell them exactly what you want and how frequently you want to get it. And here's some recipes for the next month. You can let them surprise you, by the way, or you can pick summer vegetable and egg panini with Calabrian chili mayonnaise and caprese salad. And here's another one, skillet vegetable chili with cornmeal and cheddar drop, biscuits or garlic butter shrimp and corn with green bean salad and roasted purple tomatoes. So you will get all you need to make these wonderful meals. 40 minutes or less, you can finish it, and you can do this with your family, your friends. You can impress people at what a great chef that you are. You can teach yourself how to cook just by doing this. They give you instructions on how to do everything. And it's really a lot of fun. So as I said, I hate to shop because I don't like to go to the market. And I always forget something. And also, you need a little bit of something. You end up buying a ton, and then it goes bad. And that doesn't happen here because you get exactly what you need. So I love this idea of having it delivered directly to you. Nels, have you tried these yet, or you're still working on it?
Still
working on it.
There. This sounds like, first of all, delicious. It also sounds like culinary evolution in a sense. I'm just thinking of our episode here where everyone gets the same starting ingredients. I'd love to see pictures, actually, of the product and compare notes. There might be some culinary mutations along the way. How
interesting. Yeah, if we each got the same thing and tried it and see what ends up. You know, I listened to a podcast by Malcolm Gladwell, and he was also doing a Blue Apron ad. And at the end of his episode, he did a taste test where they used Blue Apron to make something and then bought the ingredients themselves and made it separately. And a blind taste test, he said, this has got to be the Blue Apron. It's better. Isn't that cool? Anyway.
Interesting. Yeah.
With Blue Apron, there's no weekly commitment. You get deliveries when you want them. And, of course, they reduce food waste because they ship only what you need for each recipe. They have a freshness guarantee that promises every ingredient in your delivery arrives ready to cook, or they'll make it right. Check out this week's menu. Get 3 meals free with your first purchase with free shipping. Go to blueapron.com/TWiE, TWiE. That's blueapron.com/TWiE. You'll love how good it feels and tastes to create incredible home-cooked meals with Blue Apron, so don't wait. Blue Apron, a better way to cook. I always get hungry when I do these.
Yeah, really.
Blue Apron ads.
Exactly.
Maitreya, did that make you hungry?
I've actually used Blue Apron. It's great for weeks when you know you've got some big grant deadline and don't have time to do it.
Absolutely. Many people have told me, oh, yeah, I love that. And I think it's great for scientists or anybody who's busy. Yeah, you have these periods or you just get home from, being at Cold Spring Harbor for how long are you there? 6 weeks?
3 weeks.
3 weeks. And you have no food in your house, and you could arrange to have a Blue Apron box come.
Mm-hmm.
All right, let's talk about what you found.
Yeah, so back to these TY elements. What are these transposable elements? Maybe give us some background on that and yeast.
Yeah, sure. So TY elements are the yeast transposons. They are like little retroviruses that are in the genome. They're not infectious. They can't pass from cell to cell, but they can make copies of themselves and insert them in other places in the genome. So all sorts of different, pretty much nearly every organism has transposons of some sort or another, and budding yeast is no exception. And so these are really interesting potential players in evolution because these insertions can cause both loss of function mutations, where maybe they insert in the middle of a gene and disrupt it, or they can cause gain of function mutations because they're known to have a promoter element in it that can activate expression if it inserts into a promoter. So that's what we largely got, was insertions of transposons into a promoter as an activating element.
Yeah, that's cool. This is sort of coming up as a bit of a sub-theme in some of our other TWiEVO episodes. So if you remember, Vincent, I think it was episode 10, we talked about the peppered moths, this adaptation to pollution in the UK.
Right.
And it turns out that there's a recent paper in the last year linking that to the insertion of a transposable element in the ancestral moths. Yeah, it's really cool. Yeah,
it's amazing. So roughly how many transposable elements are there in your strain? Do you know?
So the lab strain has about 30 of the one family of transposons. There's a bunch of different families that are all similar in various ways, but about 30 of the full-length ones.
And so the idea is that these are coming out of the genome and just randomly inserting all over the place. And if you happen to have a phenotype you're selecting for, you'll select for the insertion at a crucial point, right?
Right, exactly. And it's also a large effect mutation. So we did a study a few years ago where we mutagenized a promoter and asked, can any single point mutation activate its expression as much as, say, just simply duplicating that gene? And what we found was that it was really hard to get point mutations that increased expression by a whole lot. And so these transposons might be an alternative way to increase expression. They're a way to get high-level gene activation with a single mutation.
That's really cool. So, Maitreya, do you have a sense of, given the 300 generations in the Chemostat, how many TY hops happen just overall?
Oh, gosh, that's a good question. So our population size is on the order of 10 to the 9th cells. And the mutation rate for transposons is, I think it's about 1 in 10 to the 6th per generation. So, but then it's hard to kind of handicap that by the copy number. But it's definitely a lot. Pretty much every generation in the Chemostat, you will have transposon insertions to select upon. So we don't think that they're necessarily like mutation limited for this. Yeah. One thing that's kind of interesting to think about is if you are the first cell to get this mutation, how does it help you? Because you can stick to other cells that don't express the flocculin proteins, but not very well. So what works best is if you have other cells that also are expressing these flow genes, and then you can stick together really well. So we think there might be some frequency-dependent selection that we have not been able to investigate yet.
But if there's one, as long as there's not a fitness cost to the insertion, then it'll divide, and then you'll have a couple.
Right. And we mix the Chemostat by vigorously bubbling air through it, but we think that there's probably inconsistencies in mixing, and so how long, if after you undergo cytokinesis, how long it takes to stir away the daughter, it could be that they stick together immediately when they divide. We haven't actually watched like one of these little cultures divide at the like microscopy level.
Now, would it be possible to remove all of these transposon insertions and start with a strain that didn't have any? Are they needed for fitness at all?
Oh, so no, the transposons aren't. And there are actually some strain backgrounds that don't have any transposons. And you could imagine engineering them all out. So, there was that paper recently where they got rid of all of the pig retroviruses, right?
That's right. Yeah.
So we saw that paper and we were like, we could do that for yeast.
Good.
Although you might end up with a strain that just has a lot of genome rearrangements. So the other way to do it is you can make mutations that lower the transposition frequency, right? The problem is some of those mutations cause side effects. So they don't just affect TYs, they affect like gene expression more generally or something like that. And we think it'd be difficult to do that. One interesting idea is that Jeff Buca's lab and all his collaborators making the synthetic yeast genome, they are systematically engineering out the transposons as they go, which is hilarious because Jeff is one of the primary people who studies transposon biology.
That's right.
Yeah. So he's systematically eliminating them. So we would love to evolve their genome and eventually see how it comes up with. So transposons aren't the only way to get the phenotype. We did find other mutations as well that are simple point mutations. So there are other mechanisms to do it, but certainly getting rid of the TYs would block off one path.
Mm-hmm. So among your 21 of the 23 clones, it sounds like this TY insertion in the promoter of flow was pretty common, but it wasn't just like completely common in the sense that the insertions could be in different places and in the element could be in different orientations. Is that true? Or how much variation is there in the types of insertions?
So it always had to be in the same orientation. And that's known because the piece that does the activating is at one end and works in one orientation. But it could be in different locations. And the flow, so this gene that we saw these insertions in is called flow1. And it's one of the big flocculin genes. And it has a really enormous promoter, actually, for a yeast promoter. And it's not totally mapped out what binds where. And so we think that the insertion profile probably does tell us something about the promoter structure, but we're not entirely sure what. But they definitely could be in different places in different clones. We didn't see the same exact location used twice.
Mm-hmm. What's encoded in the flow gene? Is that a regulator itself?
No, so it's actually one of the proteins that is expressed on the surface of the cell that causes this sticky ability. So it's one of the proteins that can actually stick to other proteins.
Got it. So this is making more of that protein. It's increasing transcription, right?
This gene would normally be activated by that flow8 regulator, which has a mutation in this strain. Mm-hmm. So the transposon insertion is pretty much a bypass suppressor of the flow8 knockout.
Got it.
Right? It's allowing activation even in the absence of the normal activator. Mm-hmm. And you can get the opposite as well. We also saw loss of negative regulators.
Mm-hmm. Of flow1.
Of flow1, right.
Okay, so those are some of the other changes. In addition to the transposon insertions, you got some others in specific genes, and some of those are, as you just said, eliminating regulation of flow1. They were negative regulators of something else, right?
Exactly. So one of those genes is called TUP1, and it is a repressor of the flocculation genes.
Okay, so you get a mutation there, and that repression is relieved to some extent,
exactly.
Right? Huh. And what are some of the others?
So we also found some mutations in other transcriptional regulators, this thing called the mediator complex, which is this huge complex. We still don't totally understand why we found mutations in subunits of it and not others. It could be just because this is kind of an unsaturated screen, right? We only looked at 23 clones, so it could be that if we did the experiment 100 more times, we'd discover more things. We did find many of the mutations only once, so it's certainly possible that there are other targets out there. But mainly, we got really lucky actually, in that nearly all of the mutations funneled through flow1. So even these regulators that regulate multiple flow genes, it turned out that they needed a functional flow1 in order to have their effect. So we did an epistasis analysis to ask, are they dependent on wild type flow1 or not? And it turned out that they all were, which was a huge amount of luck because it meant that by just getting rid of one gene, we could like wall off this whole set of mutations that can cause this trait.
So, and you mentioned that in some of the back crossing that you did, that you're able to use bulk segregating analysis to sort of identify some of these causal alleles or dissect away the flow1 from some of the other mutations that might be going on. Can you just describe that again a little bit? So you take these tetrads, which are the four meiotic products that yeast goes through on mating, and you can physically dissect those apart and then ask what traits are segregating in each individual spore. So, and what is the analysis that you do from there?
Yeah, that's right. So it's a really nice trait to look at because what we did was we saved all of our segregants from the back cross in a 96-well plate. And what you can do is just go in with a multi-channel pipette and resuspend them all and do exactly the experiment that we showed in figure 1, which is watching how the culture settles in a test tube. You can also do that in a 96-well plate. And so it's really cool. You can watch them go where half of the progeny clump really fast and settle, and half of them stay lofted and cloudy. And you can score them pretty easily just by eye. You can also put them under the microscope and see the clumps forming, which is really neat.
So because of what you found that everything seemed to be acting on flow 1, you decided to make a flow 1 null and repeat the whole evolution experiment, correct?
That's right.
And
we wanted to see if just getting rid of flow 1 would stop evolution of this trait or slow it down or at least make it rarer.
Mm-hmm. And what happened?
Yeah, so Elise and Kalina spent months of work trying to do this experiment because we wanted to really pin it down. So we did, 32 more evolution experiments of each of these because it doesn't always happen, right? It's
sure.
So we needed to do enough numbers that we could to get there, and so we did 32 of each. And of wild type intermingled with the mutant so that we would have like them all uniform and everything. And sure enough, at the end of the day, we found out that the flow 1 knockout, only 1 out of 32 of the cultures developed the trait, which we were really excited about because that's way better than what we were doing before.
Mm-hmm. So, but you mentioned before that, sort of one of the motivations of the study was to try to stop evolution, that you'd use evolution to stop evolution, but it sounds like it still, at least in one case, did kind of sneak through, or adaptation did happen. What happened in that case, actually?
Yeah, so that is a really cool clone because it got a transposon insertion in the promoter of a different flow gene. Huh. So it for whatever reason, that gene is less important in the wild type. We don't know why it's less of a target than flow 1 is. Could be something, TY elements have a preference for sequence that they can insert in more frequently than other sequences. So it could be that its promoter is less hospitable as a target, or it could be that the fitness consequence is different, or who knows. We haven't really tested that systematically, but flow 9 is next on our hit list.
I see.
We're hoping that by knocking out both of them, then maybe we can push that horizon out even longer.
Huh. So what would you predict from that experiment going into it? Will that actually stop evolution, or do you think there's still other pathways that might be exposed?
Oh, I think, what's the saying, right? Life finds a way.
From Jurassic Park.
From Jurassic Park, right? Jeff Goldblum. But so, as long as there are ways to get the job done, I think that there are other flow genes. This is a pathway that has lots of gene duplicates involved. So it could be that we at least will make the problem harder for them. So, at the end of the day, like, we are interested usually only in a few hundred generations. Enough interesting things happen in just a few hundred generations that we don't necessarily need to go for thousands and thousands to get the types of results that we're interested in. And I think the people in my lab might revolt if we tried to do all experiments at 1,000 generations instead of just a few hundred. But I think that the strain gets us to a point where we can do more of the types of experiments that we want to do, which is what the goal was.
So you in your introduction, even in the discussion, you talk a lot about modifying strains for various purposes so they cannot aggregate or form biofilms. Can you tell us a little bit about what the future of that?
Sure. So we were hoping that, by understanding more of what the genes are that are contributing to this process, that we could also just understand the pathway better and be able to control it a little bit more. And one thing that we've gotten interested in is, would different strain backgrounds that don't have this flow 8 mutation, for example, would they also rely on flow 1, or would they have kind of different ways to do this? So we've now started evolving different strain backgrounds and different species even to ask how they evolve the trait. And even diploids versus haploids might evolve the trait differently. So it could be that the flow 1 deletion is a good solution for this particular strain background in this particular ploidy, but wouldn't necessarily be a catch-all solution for everything. So that's kind of the next layer of how complex is this now that we start throwing new variables into the mix.
What other organisms have flow-like genes?
A whole bunch of them. And in fact, the genes are very well conserved with pathogenic yeast. So like Candida uses all these same genes to create, the nasty biofilms that it uses to invade tissue and to persist on hospital surfaces, things like that. So that was one of the other nice side effects of this, is that, in addition to, because how those things evolve in patient populations or in the hospital is a case where you definitely would like to try to stop evolution, right? Yeah. Now, of course, you can't go in and engineer a gene knockout in a pathogen, right? But but you could maybe use that as a drug target or something like that.
You also mentioned industrial applications of strains that don't make biofilms. What are some of the things that would benefit from that?
Yeah, so creating these kind of aggregates can kind of clog the tubing and the pipes at big industrial fermenters. And also, it's actually a trait. One of the main places where this trait's understood is in beer brewing, where bottom fermenters and top fermenters have different flocculation properties. And when strains flocculate during a fermentation run is really important to how the beer turns out. So we've actually gotten some interest from brewers on this who want to try to control flocculation better, which has been kind of fun to think about.
It would be interesting. Maybe take away flocculation and see what happens. Maybe probably make a very different brew, right?
Yeah, right. You imagine like the Hefeweizen, right, with the yeast still in the mix versus one where the yeast all settle out. It's kind of, if you think about it, nobody wants to centrifuge a giant fermenter of beer, right? Yeah. To get rid of all the yeast. So instead, flocculation is kind of the way of the yeast all clumping together, settling out of the fermenter so that you can pull the beer off and have it be still clear. So that flocculation potential is actually one of the main things that the brewers care about.
And if I'm not mistaken, sometimes there's always going to be some cells in the beer, right? Because you can't get them all out.
Sure.
And I think sometimes those are a problem in bars, when in the pipes that deliver the beer, right, from the keg to the tap, and sometimes they clog up. So.
Oh yeah.
Could be a lot of beer these days is pasteurized too, so I don't know if they would grow much, but certainly a lot of things aren't and have they have yeast in the bottle as the way of making it carbonate, things like that.
Interesting.
I'm curious as well, Maitreya, kind of comparing some of the different outcomes that you got. So not only the flocculators, but then also your the snowflakes, the aggregators.
Oh, yeah.
The mother to die. So have you done anything like competing the two of those? Do they have different niches, or would one do better than the other? Do you have a sense of that?
So that is one thing that we are still trying physically to understand how it works. So it turned out that the mother-daughter separation issue is more associated with wall sticking than it is with making clumps. So the strains will make clumps, but they're smaller clumps than the flocculation mutants. Strains that actually flocculate will make these big, robust clumps, whereas the strains which have the mother-daughter separation will make smaller clumps that take longer to settle. But what it turns out they're really good at is sticking to the walls. And we really don't understand why that is. It could be that they just have like larger surface area and are able to glom on better, or it could be that because they have something in the septum that should have been hidden by cytokinesis, it's still revealed, and that has some sort of electrostatic interaction. We really just don't know. But what that means, though, is that knocking out flow 1 did not prevent our wall growth.
I see. Yep.
So wall growth we still observed.
Yeah, interesting. I'm also wondering, are there higher order kind of interactions here? So will the mother-daughter non-separators start to stick to the flocculators in different patterns? Do you start to see kind of cooperation even emerging?
We've never mixed them together to see what they would do. Or what something with both mutations would do.
Yeah,
we don't know that. And doing the competition assays turns out to not be trivial. So the way we normally do our competition assays to really measure fitness is we'll mark the mutant strain with a green fluorescent protein, and then we'll compete it against, say, the ancestor strain, which doesn't have a fluorescent marker in it. And then we can use a cytometer to count the number of green cells versus the number of dark cells as we have them fight in the chemostat. And when you have the clumping phenotype, that means that the cytometer does a very bad job. And of course, like I said, clumping up and messing with your tubing is a big deal, and so we try not to send our clumping strains through our delicate cytometer as much as we possibly can. So doing the fitness assays is a little bit harder than usual. So we really don't have great measurements of the fitness benefit provided by this trait. Or of the kind of comparative fitness benefit. We have some information because we can kind of quantitatively measure the phenotype, like with that settling assay or with some other things that we've developed. But like you can measure surface sticking using a dye and see that biofilm develop on a surface. But we haven't been able to kind of do the head-to-head competitions that we normally like to do.
I have a really important question for my last one. And that is, what were some of those flow jokes that you mentioned?
Well, they're mostly like flow from the car commercial ad or from the old Mel's Diner. Or let's see, there are a few. The SGD website does a really good job of writing articles for a broad audience about science articles. And so it had a whole number of little things snuck into that article. I'd have to look back at it to remind myself of it, but it was nice that they covered it.
We'll have to check it out, Nels, for a title,
Yeah, well, that's what I was going to say. I'm sure that I could predict what Vincent's doing here, which is fishing for a fishing for a title for our show. A couple ideas. One that I'm currently working title I'm fond of is E pluribus cerevisiae, from many yeast become one.
Oh, yeah.
That's good. That's very good. And you have yeast go with the flow. That's a good one.
That's not bad either. Yeah.
Yeah. We'll see. If you have any ideas as well, Maitreya, for a episode title, feel free to let us know.
So for this, the Genome Database website, we would look at the original article, right? And then it would be around that discussion.
They post it on their front page, and then they have an archive of all the other ones. So it was posted around the same time as our article came out.
Okay. Got it. Cool. I'll look at that. All right. The last thing we do here is do some picks, and Nels and I. Looks like, Nels, we are sort of on the same page here.
Yeah. So we have some science picks of the week, and we'll go through this, Maitreya, if you have one. So we'll definitely link to your Cold Spring Harbor yeast course, and we could use that. Or if you think of some other links, feel free to join us here at the end. Are you actually, speaking of the Cold Spring Harbor course, are you still teaching that going forward? Or?
Oh, I just retired. This is my last year.
Yeah, retired. It's a funny word to use at your age, right? Yeah. What do you got now?
Well, we keep it fresh, right? Having new instructors come through every few years is a great way. Like when I started teaching it 6 years ago, we didn't have any sequencing labs. So I came in and developed a bunch of sequencing genomics labs, and
cool.
I felt like I've kind of asymptotically hit as good a job as I can of teaching those, and that it's somebody else's turn to try it out.
Cool. Yep. So Vincent, my pick of the week is, and I think ours are related. So thinking about just with Hurricane Harvey still unfolding, and actually some of the aftermath, and we'll get to that in a minute. But one thing that struck me, and actually with a little bit of a link to this idea of biological aggregation, actually, is that there are these rafts of fire ants that are showing up in some of the flooded areas in Texas. And so, these fire ants are an invasive species, and what they will do is, under conditions of flooding, they'll actually form these aggregates where they lock their legs together and then protect the queen and other members of the colony by floating on these mats, basically. And this is actually not a small problem that's unfolding right now, is that there are these, you can imagine if you're trying to get out of floodwaters yourself, that if you came into a raft of fire ants, that this could actually be a medical issue.
It's amazing.
Yep.
Look at that.
Yep. Yeah. So I have a couple links. One is a Wired story mentioning that they're seeing these floating mats all over the place of fire ants right now, in and around Houston, and then another YouTube video that actually shows the process. It's really, I think, a striking macro evolutionary and sort of ecological demonstration of the impact that biological aggregation can have, and how this has been part of fire ant and other insect evolution as well.
A biofilm of ants.
Yeah, exactly. That's right. That's right. Yep.
They make a good point in this article. Why didn't it happen during Katrina? Because the waters came in too quickly and all the ants died. Yeah.
And so here, with the sort of slower with the ongoing rain and the slower rise of the level, these fire ants have been deployed. I saw, I think, some other photographs of this where it's not just a single raft, but it's like collections of rafts that are starting to pile up. It's just incredible, the number of ants. Yeah. Yeah.
I wonder if they have flow genes.
So they definitely, it's not just, so part of it is mechanical, I don't know a lot about it, which is the linking of the legs, but there is like wax and cuticle that's involved in, at least, it might not waterproof the ants, but it does give them some level of water resistance to have the waxes that they're secreting also form part of that organization. And I think some of those rafts or clumps of ants are like really stable and can, get these, it's not just a few hours that they can link together in these films or rafts for many days or even weeks as they sort of recolonize or look for dry ground.
But it's also a behavioral response to water. And you can imagine that there may be some areas where you never get any flooding, and maybe the ants never do this, right?
Yeah, right. So that's, it's an interesting sort of question, I think, of how this aggregative phenotype or behavior can play out at different levels of biological organization, and how long is that evolutionary memory, in a sense, and how is that encoded? Really interesting questions. Yep. Cool. So, and so then maybe one more before I'll hand it off to you, which is, I just noticed as well, obviously keeping a close eye on the hurricane and now the tropical storm that's still churning away, both in Texas and now in Louisiana, is that just in the last day or so, the March for Science, the Houston, Texas chapter, is putting together a database of labs around the country that are offering either to host potentially displaced labs in and around Texas or related assistance. And so I've got a link there for scientists kind of coming together and trying to provide any, as I've heard, just some cases from some friends of places like MD Anderson, which is, I think, currently closed because of flooding, and other institutions that are probably going to be obviously in recovery mode for a very long time. And so some of the consequences that will have for science, for both grad students up through PIs, and how they might cobble through and sort of continue the research programs either in other places or with some help. And so I think this is a great opportunity as well for scientists to band together in another way and keep science moving forward.
Yeah, just to put that in perspective, here we had Sandy a number of years ago, and there was a lot of flooding here, and the at New York University Medical Center, they lost all their mice because the animal facility was unfortunately on the ground floor, and it flooded, and the pumps weren't working, and it took them many, many years to recover. So that's, these are real needs to have money for that, because they are up and running again, but it's quite costly.
Yeah, I agree. I know of one colleague who at the University of Houston was updating some other folks and mentioned that they had moved a lot of the mice out of the basement where a lot of those animals are housed, sort of as a preemptive measure in that case. So hopefully some lessons learned along the way, but certainly there'll be some assistance needed.
Here we have the mice in the penthouse.
I see. Good idea. Yeah. So how about you, Vincent? I think you have some related links.
Yeah, so I have a link to the Red Cross, which is going to directly go to the page where you can help people affected by the hurricane. You can choose to click buttons for different amounts of money and donate. So I think that's a really good cause at this point. And there's also a NPR link. I think you put that in, Nels, Probably, right?
Yeah, exactly. And so there it's another just kind of a charity navigator, so that you could think about donating to different organizations to kind of spread out and support folks down there who are still in some cases, it looks like a lot of trouble.
Yeah, I thought we should use at least one of our picks this week to do that. It's important. It's an important cause, so think about that. All right. That is TWiEVO 22. You'll be able to find it at microbe.tv/TWiEVO. You can find it at Apple Podcasts. And most of you I know listen on your phone or your tablet, mobile device, where you can have apps that play the podcast. And I would just suggest that you subscribe to TWiEVO. Very easy to do. You just can search for it and subscribe. It doesn't cost you anything, and you get every episode as we release them. That way you don't miss it. And it helps us to secure advertising if we can have lots and lots of listeners. And if you like what we do, consider supporting us. You can go to microbe.tv/contribute. We have a number of ways, including Patreon and other ways of giving us financial support, for which we'll be eternally grateful. And if you have questions or comments, please send them to TWiEVO, TWiEVO, at microbe.tv. Our guest today has been Maitreya Dunham. Thank you so much for joining us today. We really appreciate it.
Oh, thanks so much. This was great fun.
And yeah, thanks, Maitreya. That was great to get caught up on some of this exciting work.
We'll put a link to Maitreya's lab, of course, but you can also find her at Dunham Lab, one word, on Twitter. Nels Elde is at cellvolution.org. He's on Twitter as L Earlybird. Thanks, Nels. Good to see you again.
Yeah, you too, Vincent.
And I'm Vincent Racaniello. You can find me at virology.ws. I'd like to thank the sponsor of this episode, Blue Apron. The music you hear on TWiEVO is performed by Trampled by Turtles. You can find their work at trampledbyturtles.com. You've been listening to This Week in Evolution, the podcast on the biology behind what makes us tick. Thanks for joining us. We'll be back next month. Until then, be curious.